day. This is the Professor, CBMD PhD, and today's Wednesday, August 9th, 2023. It is 5:32 and 56 seconds, AM Eastern Standard Time, and today's research, Origins of Life. So let's jump to it here. Exploring the origins of life. A new model helps to understand the self-organization of molecules into living structures. Date August 4th, 2023. Source: Max Planck Institute. As catalytic molecules can form metabolically active clusters by creating the following concentration gradients, this is the result of a new study. Their model predicts the self-organization of molecules involved in metabolic pathways, adding a possible new mechanism to the theory of the origin of life. The results can help to better understand how molecules participating in complex biological networks can form dynamic functional structures and provide a platform for experiments on the origins of life. One possible scenario for the origin of life is the spontaneous organization of interacting molecules in the cell-like droplets. These molecular species would form the first self-replicating metabolic cycles, which are ubiquitous in biology and common throughout all organisms. According to the paradigm, the first biomolecules would need to cluster together through the slow and overall inefficient processes. Such slow cluster formation seems incompatible with how quickly life has appeared. The scientific investigators and microbiologists have now proposed an alternative model that explains such cluster formation, and thus the fast onset of the chemical reactions required to form life. And for this, they consider different molecules in a simple metabolic cycle, where such species produces the chemical used by the next one. The only elements in the model are the catalytic activity of the molecules, their ability to follow concentration gradients of the chemicals they produce and consume, as well as the information on the order of molecules in the cycle. And consequently, the model showed the formation of catalytic clusters, including various molecular species. Furthermore, the growth of clusters happens exponentially fast. Molecules hence can assemble very quickly and in large numbers into dynamic structures. In addition, the number of molecule species which participate in the metabolic cycle plays a key role in the structure of the formed clusters. The model leads to a plethora of complex scenarios for self-organization that make specific predictions about functional advances that arise for odd or even number of participating species. It is remarkable that non-reciprocal interactions, as required for the newly proposed scenario, are generically present in all metabolic cycles. In another study, the researchers found that self-attraction is not required for clustering in a small metabolic network. Instead, network effects can cause even self-repellent catalysts to aggregate. With this, the researchers demonstrate new conditions in which complex interactions can create self-organized structures. Overall, the new insights of both studies add another mechanism to the theory of how complex life once emerged from simple molecules, but more generally uncover how catalysts involved in metabolic networks can form structures. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.